Hello, Chi Hoàng. How are you? Em khỏe không Hoàng? Em khỏe không Truy?
Good evening, Dr. Lợi. Yeah, I'm good.
Okay. Are
Hi,
you ready?
how are you today?
I'm good. I'm good. Thank you.
It's nearly time for the presentation. Can I have a right to share the screen?
Sure. Please give the share permission for Miss Chi Hoàng.
Recording in progress.
Chưa đâu em ơi.
Dạ rồi thầy.
So,
Phải record cái chưa cần record. Hello Siva, how are you?
Recording stopped.
Hello.
Hello.
Hi. Hi, Lupi. Hello.
Hello.
Yeah, can you hear me?
Yes, I can hear you well.
Mm-hmm.
Can I call you Siva or Siva Kumar?
Yeah, you can call me Shiv. You can call me Shiv.
Okay, sure. Okay.
Yeah. SHIV Shiv.
Yeah. Can you please share your screen just to make sure that you can share your screen well?
Can you see my screen?
Yes, I can see your screen. Perfect. Yeah. Can you make a full screen?
Yeah.
Okay. Perfect. Yeah. Thank you.
Yes. Okay.
Yeah. And the next one will be Dr. Anna. Hello. Dr. Thana, where are you?
Anh
So
Lợi, Dr. Thana is joining. Just wait a minute.
Okay.
So the order will be first. Who's the order? How it will be?
So Miss Chi Hoàng will be the first, and then you will be the third, and then Dr. Thana will be the second.
Okay, so I will be the like last. So that's the order.
Exactly.
Yeah, sure.
Yeah.
Because I will be doing data analysis. Thank you.
Okay.
Thank you.
Yeah. So so today we will have the presentation from QIAGEN for variety of the production as well as the service. Beside the DNA extraction for tissue input or the FFPE input or the cfDNA input, the QIAGEN also have a lot of the solution kit and so on for the cancer result as well as for the clinical application. And the last part will be Dr. Siva will talking about the bioinformatic analysis for cancer as well. Yeah. So please stick until everything is finished from beginning to the last. We have totally 3 talk. Yeah. The first it comes from Miss Chi Hoàng. She will give the overview and all the solution for the DNA extraction as well as the DNA library prep preparation. Yeah. And then later we will have the solution for cancer next generation sequencing cancer genomic. And the last one we have the bioinformatic. I just repeat it again to make sure that you have the the content of the full talk of three talk today. So không biết em có bổ sung gì thêm không? Em ơi, em có bổ sung gì thêm không bên QIAGEN em?
Phần chia sẻ của anh Lợi cũng khá là đầy đủ rồi. Thì trong cái buổi ngày hôm nay thì bên em sẽ có 3 speaker, thì các bạn cũng đã chọn đủ rồi đó. Dạ.
Ok.
Dạ.
Cái bạn Thanas không biết là có thể xem thử á, cho một lát cho nó trơn tru á em, trong lúc mình chờ đợi mấy bạn.
Yeah, Thanas, can you share the screen, so we just want to check if everything good now?
All right, just a moment.
Okay.
Just a moment. Can you see my screen?
Yes, I can see your screen. Can you make
Right.
a full screen, please?
Sure.
Just to make sure that on the right side of it. Okay, perfect. Yeah, thank you, Thanas.
No problem.
So we have 40 minutes for each talk, and please keep your time. So it's better to have 30 minutes and then have 10 minutes Q&A after the talk. And the last part we will have like 20 minutes to discuss. Yeah. So stay still until we
Tiến sĩ Ngô Hữu Lợi, Đại học New South Wales.
Okay, so now we're ready for the first
Recording
talk.
in progress.
Okay.
Mr. Chiu Han, can you make a short introduction for yourself first, and then you will go on your presentation, please. Thank you.
Yeah, okay. Thank you, Dr. Lợi. My name's Chi, and I'm an application specialist at QIAGEN. Yeah, and today it's my pleasure to have a talk about high quality nucleic acid extraction from challenging oncology samples. To be sure that everything will work well, I would like to check whether can you see my screen?
Yes, I
Yes.
can see your screen.
Okay, now I turn to another screen. Can you see it?
Yes, I can see it.
Okay, that's well. Okay, so in today's topic, I'm gonna cover 4 parts. The first one is about an overview about cancer and cancer genetics, and then some challenges and solutions for FFPE and liquid biopsy samples. The last one will be some publications. Okay, so let's go. Let's talk about cancer and cancer genetics first. I'm sure that all of you already know about cancer, right? Cancer is a disease caused by uncontrolled cell growth and division, and it can originate in almost any tissue or organ and may spread to other parts of the body. And until now, cancer is still a burden worldwide. You can see the graph here on the left, on the right, the number of cancer cases, new cases, increasing dramatically, and it is expected that by 2050, 35.3 million new cancer cases will be recorded. And you can see the number of deaths here is increasing too. And by 2050, it's predicted that 18.5 million deaths. And you can see that the number is really much huge, right? So there will be a need to have early screening, diagnostics, and like treat cancer. So to treat it, you must know what is the root cause. And 5 to 10% of cancer are caused by inherited genomic variants or germline mutations, and the rest of them are caused by acquired genomic variants. So regardless of cancer variants or cancer types, its root cause is from the genome of cancer, right? So to treat it efficiently, we must know the genome of cancer. That's why cancer genomics is get a lot of interest recently. Cancer genomics is the study of totality of DNA sequence and gene expression differences between tumor cells and normal host cells. And it can help clinicians in various aspects. You can see the tables here. Okay, I make some kind of the benefits of cancer genomics, and here is the example, some example. For example, cancer, if you know the genome of the cancer, you can do the screening for early detection. For example, for retinoblastoma, if children have a history, they can do some kind of a screening test for RB1 mutation. If the tumor is detected early, it can be save the children's eyes and some useful vision. Otherwise, the eyes may be removed. Okay, and here, with cancer genomics, it can help clinicians to diagnose or give some prognostics. For example, PCR, ABL for CML. When you have the diagnostic, you can have the treatment just for personalized treatment. For example, not all patients with gastrointestinal trauma tumor can be benefits from some kind of medicine. It's just for some of the patient with this kind of tumor and have the mutation of c-KIT, PDGFRA, BRAF can be benefits from the suppressor drug. And it can also be used to monitor the treatment response and recurrence. For example, in APL, the mutation PML-RAPA can be used to monitor the treatment and as well as for early detect of recurrence. There are some technique to get access to the cancer genomics, but NGS has been gaining the most use, because it offers a comprehensive and efficient way to analyze the genetic changes. Here is a general workflow of NGS. First, the sample is collected and isolated. And then after having the library constructed and enriched, it will be run on NGS machine and the data will be analyzed and interpreted. Okay. In this talk, I just focus on sample isolation or sample extraction. Okay. The rest will be covered by Dr. Thanat and Dr. Shilf. Okay. To get to know the cancer genome, the first step and the most important step is to gain the DNA or RNA of the cancer sample. So here are some samples typically used in oncology. The first one is FFPE tissue. It is commonly used in Vietnamese hospitals. And the second one is fresh frozen tissue, often used in research. And the one that is on trend here now is liquid biopsy. And next is bone marrow aspirate. It is used in leukemia diagnostics. And the final one, if the resources is limited, some doctor can do fine needle aspirates. And while FFPE tissue is considered to be a standard and liquid biopsy looks promising, dealing with FFPE tissues and liquid biopsy is not easy. So today I'm going to give you some challenges and how we deal with this kind of difficulties. First, let's look at FFPE. Here I present you 5 challenges when you work with FFPE tissue, FFPE sample. First, FFPE is precious and irretrievable sample, so you cannot afford a waste. And the third, for the second and the third, you can see the pictures on the right here. If the tissues is it's like fresh or frozen, it doesn't go through the process of fixation. After the extraction, the DNA can be released and will not be fragmented. So the quality as well as the quality is really good, right? You can see here. However, if it goes through the formalin fixation, it will create some cross-link. You can see the line, the black line here. The cross-link between the protein and the nucleic acid, between the protein together and between the nucleic acid together. And after the extraction, some protein still may attach to the nucleic acid. So it's affect the quality, so might interfere with downstream analysis. Or you can see here, because of the process of fixation, the DNA is not in integrity anymore. It is fragmented, and actually highly fragmented nucleic acids. And if the process is prolonged, you can see here, the quality as well as the integrity of the nucleic acid is not qualified enough for the molecular applications. And next, it's about the low use of DNA, RNA, and protein. And finally, it may pose a potential that it may give inaccurate genomic or transcriptomic and proteomic data. The question is why? You can look at the picture here at the bottom. You can see that the gDNA before fixation here, CG, see? But after the process of fixation, C turned to U because of process called cytosine deamination. C turned to U. And after PCR amplification, U will bind to A, and A will bind to T, and finally, it's not CG anymore, it's TA now. So it will cause false positive for low frequency mutations when you use NGS to detect them. Okay, so you see, there are lots of difficulties to deal with FFPE samples. So how can we tackle it? Don't worry. QIAGEN offers some solutions. The first one is sample collection to dealing with the sample from the beginning. To make sure everything works well from the beginning. Okay. PAC gene tissue system if you use packaging tissue system, you may overcome all the challenges that the FFPE tissue causes. It's a formalin-free presentation, but still have the preservation of morphology and biomolecules. And also, one more thing, that if you store the sample at -20 Celsius degree to -80 Celsius degree, the tissue is not frozen. So it still preserves the tissue morphology, even with really low temperature. If you are interested in RNA, QIAGEN offers packaging bone marrow RNA system. But how good it is? You can see the graph here. It is a graph comparing between the mRNA level when stored at day 0 and day 2. After storing at day 2 and day 0, how stability it is, how stability the RNA is when compared to packaging. When the sample, the bone marrow, is collected and stored in packaging and EDTA tubes. So you can see that after 2 days, all the RNA here show minimum changes. While here with EDTA tube, it shows a significant differences, right? So packaging bone marrow RNA system is a good choice for you if you are interested in RNA. And after the sample collection, we go to sample extraction. Here on the left, I present you again 5 challenges of FFPE samples. And here is the way that QIAGEN deal with it. For the first one, for precious samples, QIAGEN allows you to simultaneous purification of DNA and RNA. So instead of do the DNA and RNA extraction separately, you can do it together at the same time. It will save your time as well as your amount of sample, right? And the cold DNA RNA extraction also allow reliable comparison of genomic and transcriptomic data. The second one is formalin cross-linking. QIAGEN, the paraffinization solution, is the best way for dealing with it. And more than that, QIAGEN provide optimized procedure. How much input volume, how many temperatures, how long could we incubate, and some specializes. I will talk about it later in detail. For fragmented nucleic acids, QIAGEN also give you optimized procedure, how to treat the FFPE. I will talk it later in detail too. For low use, QIAGEN give the kit with the high recovery efficiency and the ability to eliminate inhibitors, so you can improve the use. And for the inaccurate genomic data, QIAGEN offer treatment, and I also go in detail later. And with all of the features here to overcome the challenges that FFPE given, QIAGEN have several kits for you to choose from. And they all offer all the solutions, except for the treatment just available for KM-DNA FFPE tissues. And if you want to do the co-extraction, you use on-prep. RNA, you use on-easy FFPE tissues. And if you want to target the small sizes of RNA, For example, smaller than 70 nucleotides, you can use mRNA FFPE kits. And all of the kits can be do manually or automatically. It's your choice too. If you want to do automatically, I'm sorry, you can use EZ2 or Connect. I will show you the pictures of these machines later. Okay, now as I promised, I will give you some detail into some of the technique that QIAGEN use. Sorry, I have a cold. Okay. Looking here is the key steps in nucleic acid preparation to prevent cross-linking and use, improve use and eliminate cross-linking. The first step is the paraffinization. It will involve using hydrophobic solvents to dissolve and remove the paraffin. So when you read that, you think it's really easy, right? But if you look carefully, you will see. I will give you something that QIAGEN do do it to make it more efficiently. You know, if this process is not you cannot remove the paraffin effectively, it will affect the downstream, or maybe you cannot use the acid nucleic you extracted anymore because it's not qualified enough. So how to make it efficient? First, season QIAGEN really specific on how we treat the FFPE tissue. The first two slices must be disposed because it is usually oxidated, and it will cause highly fragmented nucleic acid. And after you have the slices, you must do the extraction immediately, because the longer you keep the slices outside, the higher extent of the fragment will be severe. Okay. And next, which is really important, is the sizes of the slices. After researching, QIAGEN discovers that 5 to 10 microliter of thickness of the slices is the best. Thicker slices will cause low yield. So if you use QIAGEN, you already have all the information to deal successfully with the deparaffinization. Okay. And after this step, protein K treatment will be given to digest cross-linked proteins and solubilize nucleic acids. And after this, the heat incubation will be applied to break cross-links. You see here, break the cross-link without inducing nucleic acid fragmentation. This step looks easy too, but it's not. If you use too high temperature, it will damage the nucleic acid. But if you use too low, temperature is too low, the cross-link will not be broken enough, and it affects the quality of the nucleic acid. So QIAGEN discovered that for gDNA, 90 Celsius degree for 1 hour or 80 Celsius degree for 4 hours are the best. Okay, and for the last two steps, QIAGEN offer all of the reagents. But for the first step, customer must prepare the solvent themselves. And to prevent the toxic and improve the efficiency, QIAGEN offer QIAGEN deparaffinization solution. And here is the graph showing the how good the solution is, the solution provided by QIAGEN. You can see here when comparing the yield between as well as the CQ when using the paraffinization solution. Dark blue, the dark blue here represent for the paraffinization solution from QIAGEN, and the light blue here represent for the melt traditional melt method. And you can see that the yield is always higher either of the method of calculation, and the CQ here is lower, much lower compared to the traditional method. Okay. Next is how QIAGEN sample extraction tackle with the inaccurate genomic data. Again, you see this picture. You see that in the fixation, during the fixation, the C randomly turned to U. And finally, it's not CG anymore, but it's TA. And it poses a threat of false positive for low frequency mutations, right? So to deal with it, on treatment will remove U here, uracil. It will remove it. So, and here is the result. Comparing between with on treatment and without on treatment. The number of mutations is recorded. And you can see that with the on treatment on treatment can remove over 90% of low frequency novel mutations. See here, that are most likely to be artifact. Okay. So that's all about the FFPE challenges and solutions. So for a summary, packaging for sample collection. Kits can be done manually or automatically with on treatment to remove artifacts. QIAGEN deparaffinization solution combined with optimized buffer, temperature, incubation time for improving use, cross-linking, and fragmentation. Finally, to save time and sample, DNA and RNA co-extraction is a good option. Okay, so now we're gonna move to liquid biopsy, some challenges and solutions. I think you are familiar with the word liquid biopsy. Okay, liquid biopsy is a minimally or non-invasive method. And it can be blood, serum or plasma, urine, saliva, or CSF, right? And in cancer, liquid biopsy will complement tissue biopsy to overcome some limitations. For example, the availability of tumor tissue, hard-to-reach tissue, for example, brain or liver. And liquid biopsy can be easier for regular monitoring to have some insights from heterogeneous tumors. Okay, and for liquid biopsy, you can work with cfDNA, cfRNA, EV-CTCs, and proteins. But today, we just focus on the first two. When working with liquid biopsy, there are some challenges that you may encounter. The first one is low and highly variable concentrations. The cfDNA in human healthy individuals is ranging from 1 to 50 nanogram per milliliter of blood. And it also have a potential for experimental bias due to the contamination with cellular DNA. And the third one is the short life of cfDNA. It's just 15 minutes to 2.5 hours in blood and maybe shorter in urine. And the fourth one is the PSA inhibitors. So to work with liquid biopsy, it requires a workflow that can avoid release of cellular nucleic acids. So it can improve the number 2 and number 3. Enable highly efficiency large volume nucleic acid extraction. It will help to deal with the first challenge here. And avoid the fragment size bias to improve sensitivity. Again, QIAGEN will give solutions from the start by sample collection. People can use PACGene plus cfDNA tube. It gives unique formaldehyde-free and non-cross-linking stabilizing chemistry, so it's not affected cfDNA. And you can see here in the graph here, it compared the stability of cfDNA. And the PACGene is in blue. They compare after storage of day 0 and day 6. At day 0, you can see that the cfDNA here, the peak here, nearly 200. It's about 107 T or 180. At day 1, for EDTA tube or PACGene, they nearly similar. But for day 6, you can see that PACGene still give the similar peak here, right? But the EDTA is have a really high peak, and it is the proof of the process of release of gDNA, cellular gDNA. And for RNA, PACGene plus RNA tube can help you with that, to help you to stabilize the RNA even at room temperature. Here also the graph on the left comparing the stability of RNA stored by EDTA and PACGene plus RNA tube, and you can see the red line is the mean value between day 1 and day 3. For PACGene, it's nearly unchanged, right? But for EDTA, you can see that after the first day, it's decreased significantly. Urine is an interesting sample, but it's difficult to be stabilized. And PACGene, QIAGEN offer PACGene urine liquid biopsy set. It can stabilize the cfDNA by minimalized gDNA release and bacterial growth. And one more thing, the additive in the tube is non-cross-linking, so it does not affect the cfDNA. And you can see the graph here. It can prove the stability of cfDNA when collected and stored in PACGene and the unstabilized urine. You can see that after 72 hours, urine stored in pathogen show the level of cfDNA nearly unchanged here. However, for the unstabilized urine here, it decreased significantly, and at 72 hours, it's nearly zero. The cfDNA is nearly zero. Level is nearly zero. Okay. After the sample collection, it is sample extraction. QIAGEN offer both manual and automatic. And QIAM circulating nucleic acid kit is considered to be a gold standard in extracting cfDNA. And now QIAGEN also have another option for you, the QIAM Mini Elute. It combines both magnetic bit and column method together, and it allows you to extract up to 10 milliliters of sample. As you remember that one of the challenges of liquid biopsy sample is the low and high variability of concentration, right? So QIAGEN offer a really high input volume, 10 milliliter of input sample, but so it can increase the yield, right? But QIAGEN also offer a small elution volume. It's just 20 microliter, so it also can improve the concentration. Okay. And for if you want to do the automatic, you can choose the 24 samples format by EZ2 Connect, or you can choose the 96 sample format by QIAsymphony. It's up to you. And here are some, I show you some automatic purification machine. This is the EZ2 Connect. You can go up to 24 samples. And here you can refer to it to see what type of sample can be used on these kind of machines. This is QIAGEN Connect. This one, the EZ2, it uses magnetic bit. But for the QIAGEN, it uses column. QIAsymphony also use magnetic bit too. Okay. Now the last part is about some publications. Here is an evaluation of commercial kits for purification of circulating free DNA. And you can see the graph here. This team, they compared 4 competitors with QIAGEN, and they have a conclusion that QIAM Menu Elute cfDNA is the best kit to give the best DNA concentration, quality, as well as the yield, compared to Promega, ABI, and another competitor from Canada. And for this one, it compared 14 commercially available kits to QIAGEN. 14 commercially kits, and they have a conclusion that the most suitable for FFPE tissue was on-prep by QIAGEN because of its yield, quality, and ability to purify both RNA and DNA from the same sample. And this one, I just give you a look at how researchers do with the urine. For urine, they use KM elute and they extract 10 milliliter of urine. And for this paper, they use an automatic equipment, Symphony. Okay. So that's all that I want to impart. Thank you for your attention. And if you have any question, please ask.
Thank you, Chi Hoàng. Thanks a lot for your nice introduction for variety of solution for the cfDNA as well as the FFPE sample input. Because my student is easy for them to ask you the question, so maybe we use Vietnamese. Sorry for boss guy. Chắc là mình sử dụng tiếng Việt các mọi người cứ hỏi thoải mái ha. Đó, ở đây chắc là Chi chắc cũng sẽ dễ để trả lời cho mọi người hơn ha. Còn cái phần sau của hai bạn nước ngoài kia thì một lát mình cứ hỏi tiếng Việt ha. Nếu mà tôi trong khả năng của tôi có thể dịch được thì tôi sẽ dịch, còn nếu không đây có mấy bạn bên QIAGEN đó và mấy bạn bên chỗ của công ty của Chi cũng sẽ giúp mọi người ha. Đó, mọi người đừng có ngại đó. Rồi thì mọi người cũng biết là mình làm bây giờ là kỷ nguyên của liquid biopsy là sinh thiết lỏng, đúng không? Sinh thiết lỏng ở đây là cái cfDNA, ctDNA đó mọi người. Đó thì bên QIAGEN là chúa tể của những chiếc nhẫn về tách chiết. Đó, mọi người cũng biết rồi đúng không? Các giải pháp về tách chiết thì bạn Chi đã trình bày rất là cụ thể. Đó, tất cả những cái thông tin và những cái giải pháp về tách chiết cho mẫu FFPE là mẫu mô đó, sinh thiết đặc đó. Thì ngoài ra cũng có CF, CT hay mọi người cũng thấy đó. Thì đây không biết có anh chị nào hỏi không? Trong quá trình chờ đợi câu hỏi của các bạn á, thì chắc tôi hỏi màu đầu trước ha. Em ơi, cái trip vaccine á, thì cái trip đó thì nó có lợi ích gì hơn so với là cái trip mà bình thường để mình có thể thu giữ và tách chiết cfDNA không em? Có nghĩa là nếu mà giờ tôi sử dụng một cái trip loại khác thì có bị ảnh hưởng gì không? Hay là phải sử dụng cái chip đó thì nó mới có lợi thế, một cái ưu điểm gì đó mà tôi ở đây tôi không biết. Có cái loại chip khác nhau thì nó cũng sẽ ảnh hưởng tới cái việc mình thu nhận cái cfDNA đúng không em?
Dạ, không bắt buộc là anh phải sử dụng vaccine thì anh mới có thể làm cfDNA. Tuy nhiên là đối với những cái túp, ví dụ túp bình thường như EDTA á, thì về cái phần bảo quản của nó phải khắt khe. Ví dụ như là anh có thể thấy là cái độ ổn định của CFD này nó không có được cao. Thì vaccine thứ nhất là nó giúp ổn định cái CFD này mà nó không có sử dụng những cái hóa chất mà cross-linking với lại CFD này. Cái nữa là cái nhiệt độ bảo quản, anh có thể bảo quản nhiệt độ thường. Ví dụ như anh thu mẫu ở trong EDTA đi thì nhiệt độ bảo quản anh phải bảo quản đúng nhiệt độ lạnh chẳng hạn, thì những cái nhiệt độ đó nhiều khi mình vận chuyển nó sẽ không có đảm bảo. Đó, thì cái vaccine này nó cho phép anh bảo quản nhiệt độ thường. Rồi cái tiếp theo nữa là cái vaccine nó có thể giúp một cái phần rất là quan trọng, đó là anh có thể thấy ở trong cái phần này, cái phần challenges của liquid biopsy á, là anh phải tránh cái bias khi mà mình bảo quản một cái quá trình lâu á, thì nó sẽ giống như là cái gDNA thì cellular thì trong tế bào á, nó ví dụ tế bào chết lập trình nè, hoặc là nó vỡ ra do bảo quản không đúng hay gì đó, hoặc là sau một thời gian thì nó sẽ vỡ ra thì nó sẽ phóng thích cái gDNA đó. Thì cái gDNA đó nó sẽ làm loãng đi cái cfDNA của mình và nó gây cái khó khăn khi mà mình phân tích. Thì khi anh có thể nhìn cái biểu đồ ở đây này, thì mình thấy là sau khi mà cái quá trình mà lưu trữ thì đối với vaccine thậm chí nhiệt độ thường cũng không sao hết, nó mức độ cfDNA nó vẫn là một cái peak y chang như cũ. Nhưng mà đối với EDTA thì nó đã bị nhiễm cái DNA rồi.
Nhưng mà tại vì trong đây em toàn là để nó RNA không.
Dạ, đây DNA đây ạ.
Là cái
Chắc
biểu
tại
đồ
em
của
nói
em
nhanh
coi
quá.
nha, plus RNA rồi.
Dạ, đây là RNA, còn cái này là DNA ạ.
Ừ.
Cái bên phải là DNA, còn cái bên
Có
trái
nghĩa là
là RNA.
nó vẫn tốt cho DNA, mấy cái CC hoặc là CF DNA đúng không?
Dạ.
Ừ. Ok. Rồi mọi người cũng thấy đúng không? Chương này không phải là không có bằng chứng, người ta cũng cho mình thấy được là cái việc mà sử dụng cái chip nào đó để mà mình có thể thu nhận mẫu máu để mình làm sinh thiết lỏng, nó cũng ảnh hưởng tới cái chất lượng của cái DNA của mình làm ha. Cái này cái lưu ý. Rồi mời bác sĩ Nguyễn Huy Thịnh, em hỏi đi em, rồi xong tới đó tới bác Nguyễn Anh.
Rồi dạ.
Em là bác Thịnh.
Thì cho tôi hỏi một cái là trong cái vaccine lúc nãy bạn có nói là một cái giải pháp mà không có dùng formalin, không có cố định formalin cho mẫu mô á, thì
Dạ.
cho tôi hỏi, cái mẫu mô ở đây mình sử dụng là loại mẫu mô gì, nghĩa là mẫu mô tươi sau khi sinh thiết lấy ra, à, và hay là một cái loại mẫu mô khác. Với cái thứ hai á, là trong cái bài của bạn trình bày có nói là sau khi cố định bằng cái patch đó thì có thể dùng cho những cái phân tích sau đó, thì cho hỏi là những cái phân tích sau đó là những cái phân tích gì, nghĩa là ngoài cái chuyện mà tách chiết DNA ra ấy, và khi mà cố định bằng cái patch này là còn có thể dùng cái mẫu mô đó để nhuộm HNE hoặc là các cái phương pháp nhuộm khác, hóa mô miễn dịch vân vân, nhuộm đặc biệt đó để mà khảo sát được cái hình thái học của mẫu mô đấy hay không, hay là khi đã cố định bằng patch rồi là chỉ có dùng để khảo sát gen được thôi. Cảm ơn ạ.
Dạ. Cảm ơn câu hỏi của bác Thịnh ạ. Thì đối với cái vaccine tissue system này á, thì sau khi mà cố định xong nó sẽ dùng mẫu mô tươi ạ. Mẫu mô tươi, và mình vẫn có thể nhuộm được ạ, tại vì nó không có thay đổi cái hình thái học của tế bào ung thư. Sau khi mình làm xong mình vẫn có thể nhuộm được.
Vậy thì á, cái quy trình mà nhuộm á, nó có khác gì so với cái quy trình nhuộm bình thường, tại vì hiện tại không, tại vì cái quy trình hiện tại là nó sẽ ừm, sẽ tại vì thường là nó sẽ cố định khối nến mà đúng không, thường là đóng trong khối
Dạ.
nến, thì là khi mà nhuộm nó sẽ có các bước là khử nến rồi nhuộm màu vân 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 vân, thì cái này là không cố định trong khối nến nữa, thì khi mà muốn cắt ra làm tiêu bản nhuộm slide thì có bắt buộc phải cố định trong lại, nghĩa là cố định lại trong paraffin hay không, hay là
Dạ, chắc câu này là bác cho em xin trả lời sau được không ạ? Tại cái phần nhuộm như thế nào em chưa tìm hiểu ạ.
cảm ơn chị.
Dạ rồi, em cảm ơn bác Thịnh ạ. Em mời bác anh Nguyễn.
Mời bác Anh Nguyễn.
Dạ vâng ạ, em có xin phép hỏi một số câu hỏi như này. Thứ nhất là trên cái slide này chị, thì ở đây chị có một cái package nữa là vaccine bone marrow này. Cái này chỉ dùng cho bone marrow với cả RNA thôi ạ? Hay là mình có thể dùng cái hệ thống này với cái tuyếp đựng mẫu này với các loại mẫu khác nữa?
Dạ. Theo đúng cái tên đó là đang dùng cho bone marrow thôi ạ, và cho lưu trữ RNA ạ.
Tức là mặc dù mình có thể dùng RNA nhưng mà mình sẽ không thể dùng với các loại mẫu khác đúng không ạ?
Dạ.
Các loại mẫu khác thì mình sẽ dùng cái tissue system này.
Dạ. Hoặc là nếu mà mình muốn dùng cho cfDNA thì bên này sẽ có cái phần liquid lỏng á.
Vâng, không, em sẽ quan tâm đến phần FFPE thôi. Cái thứ hai cho em hỏi một xíu ạ. Là như cái phần chị nói nó có cái chuyển đổi từ she sang you, cytosine sang uracil ạ, thì là mình nói là sau để mình tránh cái lỗi đó trong quá trình mình pizza lên thì mình sẽ loại bỏ cái u đi. Thế thì chị loại bỏ bằng cách nào ạ? Tức là bằng hóa chất hay là mình
Dạ, bằng hóa chất ạ.
bằng hóa chất.
Ung treatment á, cái này là một cái hóa chất. Ung là cái tên viết tắt thôi ạ. Cái hóa chất có tên ung á. Thì mình sử dụng cái hóa chất này mình ủ với nó thì nó sẽ remove, nó sẽ cắt những cái uracil đó thôi.
Vâng, tức là mình sẽ dừng ngay ở cái bước đó rồi sau đó mình mới pizza lên thì nó sẽ không bị lặp lại cái đó nữa đúng không ạ?
Sau khi mà mình dùng ung treatment xong mình phải tách chiết nè, thì mình sẽ tiến hành tiếp các cái bước tách chiết tiếp theo thôi.
Vâng, em có một câu hỏi nữa về cái phần chị nói là chị sử dụng một cái đề paraffin khác và chị so sánh là nó tốt hơn hẳn so với cái traditional method ấy ạ. Thì là bình thường thì em cũng chưa có làm thực tế nên em muốn hỏi là cái traditional method ở đây là mọi người đang dùng gì khác nhau về bản chất, về mặt hóa chất hay là có phương pháp gì khác đúng không ạ?
Ừm,
Bình thường là mình
cái
loại paraffin
bình
bằng cái
thường
len hoặc là
Dạ đúng rồi, bình
vâng.
thường thì mình dùng xi hoặc là những cái chất mà kỹ nước. Ừ.
Vâng.
Dạ.
Thế thì
Thì
tức là cái deparaffin của mình sẽ khác gì ạ?
nó không dùng những cái hóa chất độc hại này.
Ừm,
Còn dùng cái hóa chất như thế nào thì cái này là bên công nghệ của hãng nên là em cũng không biết.
tức là cái này tại vì là em thấy là mình nhấn mạnh về cái điểm là nó tăng rõ rệt về cái hiệu quả. Thì cái này nếu mà mình nói là chỉ là hóa chất khác để giống như rất là nhiều hãng họ đưa ra những cái giải pháp deparaffin chỉ là nhằm mục đích là giảm cái sự độc hại với người sử dụng thôi, chứ không phải nói về cái việc là hiệu quả của cái việc deparaffin nó có thể giống nhau, nhưng mà chỉ khác nhau là mình biết là cái xy lanh rất là độc hại cho người
Thứ
làm.
nhất là nó độc hại, thứ hai nữa là cái khả năng mà nó phá cái paraffin đó, nó được tốt hơn.
Vâng, ok chị. Vâng, em có một câu hỏi nữa là như chị nói là mình sẽ với cái bộ kit QIAGEN của mình thì mình dùng với cái độ dày từ 5 đến 10 micromet thì sẽ là tốt nhất đúng không ạ?
Dạ.
Trên thực tế thì bọn em làm thì bọn em có chuẩn bị mẫu cho bên họ làm về các xét nghiệm về gen, thì có những cái mẫu mà mình sẽ không thể cắt được nữa bởi vì nhiều khi là mẫu nó quá nhỏ, trong quá trình mình thao tác cắt nó có thể sẽ bị mất đi. Thế thì thường là họ cũng có một cái phương pháp nữa là họ lấy thẳng cái mẫu đó ở trên cái khối nến. Thế thì
Ừ.
nếu mà việc lấy thẳng trên khối nến đấy thì nó sẽ không đảm bảo được cái độ dày đấy, thì mình có gặp những trường hợp đấy mình có khuyến cáo gì, mình phải xử lý nó như thế nào không ạ?
Ừ. Nếu như mà nó dày quá không cắt được nữa thì có thể là mỏng quá, cũng có thể là dày quá, đúng không bạn?
Vâng, đúng rồi ạ.
Nếu mà dày quá thì dù bạn có bổ sung thêm cái deparaffinization đi chăng nữa hoặc là ủ thêm thì cái hiệu suất của nó cũng không cao. Thì mình phải chấp nhận cái rủi ro thôi. Chứ còn hãng đã nghiên cứu là lớn hơn 10 lớn hơn 10 micrometer thì nó sẽ bị ảnh hưởng tới cái yield, nó sẽ bị thấp. Dù có tăng cái deparaffin solution lên thì nó cũng không có giải quyết được vấn đề.
Chị có thể giải thích lại giúp em một xíu về cái phần deparaffin của mình đúng không ạ? Em thấy mình là gồm có cả 3 bước, gồm cả deparaffin xử lý bằng enzyme, sau đó là dùng nhiệt đúng không ạ?
Dạ. Cái bước đầu này là cái paraffin, cái lớp paraffin ở ngoài là mình sẽ loại bỏ nó đi nè.
Vâng.
Cái lớp màu trắng trắng ở ngoài mình sẽ loại bỏ nó đi thì nó sẽ phơi ra những cái crosslink này. Sau đó thì mình mới dùng proteinase K, thì lúc này proteinase K nó mới tấn tro cong vào trong được. Thì nó sẽ phân hủy cái protein này đi. Sau đó mình mới dùng nhiệt để mình bẻ gãy những cái crosslink giữa cái DNA và các cái protein này để mình thu được cái nucleic acid tinh sạch.
Tức là sau đấy mình vẫn sẽ phải có một bước tinh sạch nữa đúng không ạ?
Đúng rồi, đúng rồi.
Vâng rồi, em rõ rồi, em cảm ơn chị.
Rồi, cảm ơn bạn.
Rồi, cảm ơn bạn anh Nguyễn. Có câu hỏi của bạn Trần Bình Hân hay Trần Bình Hùng không biết. Trong y khoa solid tumor ví dụ như ung thư tủy, ung thư dạ dày, ung thư gan thì có dùng kết quả của liquid biopsy cho chẩn đoán bệnh trong thực hành lâm sàng hay không, hay chỉ dùng trong nghiên cứu mà thôi? Thì đây tôi không phải bác sĩ mà mấy big speaker đây cũng không có bác sĩ, nhưng mà ở đây tôi thấy audience của tôi có mấy anh chị là bác sĩ, ví dụ như bác sĩ Minh Thắng ở bệnh viện 108, bác sĩ di truyền nè, rồi tôi thấy có bác sĩ Minh Phạm, tôi thấy có bác sĩ Tô Kim Sang, đó là bác sĩ chuyên về ung thư phổi đó, và có bác sĩ về bác sĩ Nguyễn Huy Thịnh, đó là có Nguyễn Anh cũng với lại Nguyễn Huy Thịnh là cũng bác sĩ về giải phẫu bệnh đó. Và mấy anh chị em có thể giúp tôi trả lời câu này được không mọi người? Ngoài ra tôi biết được có nhiều anh chị cũng bác sĩ ở đây nhưng mà tôi không có thấy, tôi không biết đó mọi người, đó bởi vậy là mọi người thông cảm mà tôi không có liệt kê hết. Thì câu hỏi của bạn Trần Minh Hùng á, thì mọi người có thể giúp tôi trả lời được không ạ? Là cái solid tumor ví dụ như ung thư tụy, ung thư dạ dày, ung thư gan thì có dùng kết quả của liquid biopsy cho chẩn đoán bệnh trong thực hành lâm sàng hay không, hay chỉ dùng trong nghiên cứu?
Em xin phép trả lời câu này luôn tại vì nó cũng là cái làm
Dạ.
thật ra là trong các cái guideline hiện tại á, chưa có một cái guideline nào là đồng thuận cho phép sử dụng ctDNA để mà chẩn đoán các loại ung thư solid tumor hết. Tất cả các chẩn đoán xác định về solid tumor ung thư thì đều cần phải có bằng chứng của mô bệnh học để có bệnh đó. Thế nên là ctDNA nó không có phải là một cái công cụ để giúp là khẳng định xem là bệnh nhân đó có ung thư hay là không có ung thư. Nhưng mà ctDNA mặt khác thì nó có một số các cái vai trò trong cái việc ví dụ như là khảo sát những cái mà đột biến trong trường hợp mà tiếp cận đến cái mẫu mô nó khá khó khăn, hoặc là cái mẫu mô mà đã sinh thiết ra để mà làm những cái chẩn đoán phía trước đó nó còn lại rất ít, bị hạn chế, vân vân. Hoặc là khảo sát những cái ví dụ như là đánh giá cái MRD là cái lượng tồn dư tối thiểu, có nghĩa là bằng cách là khảo sát cái tính động của ctDNA. Ví dụ như là một cái gen nào đó, một cái đột biến gen nào đó mà sau một thời gian điều trị nó giảm xuống và đến một thời gian sau nó tăng lên, thì có thể là mình cần phải có những cái nghi ngờ và những cái biện pháp kèm theo. Và ngoài ra thì cái ctDNA nó cũng có thể giúp gọi là hỗ trợ thôi trong cái việc mà gọi là early detection, nghĩa là phát hiện sớm. Tuy nhiên đó, nó chỉ tất cả cái vai trò của nó, ngoài cái chuyện trong một số cái ngữ cảnh xác định là sử dụng cái kết quả ctDNA để làm một cái bằng chứng cho việc phát hiện đột biến gen nhằm mục đích điều trị, thì các cái ứng dụng khác ví dụ như là phát hiện sớm hoặc là khảo sát tồn dư ưu tối thiểu thì là cần phải được sử dụng kết hợp với các cái phương pháp khác. Do nó chỉ có cái vai trò là mang tính chất là gợi ý thôi. Dạ, vâng, thì em xin phép.
Cảm ơn bác sĩ Thịnh. Không biết có ai có thêm ý kiến giúp trả lời cho câu hỏi bạn Trần Minh Hùng của bạn. Mời bác sĩ Hương Trúc. Em có ý kiến không em? Đừng có ngại, mọi người biết thì mình giúp người ta thôi chứ ở đây không có tiền thiệt, nhưng mà có tấm lòng. Rồi, nếu mà không có ai bổ sung thêm gì nữa thì cảm ơn Chi Hoàng rất nhiều. Cảm ơn bác sĩ Thịnh và một số anh chị em đã có câu hỏi. So the next part will be the talk from Dr. Nathan. Please share your screen, please.
Yes, give me a moment. Can you see my screen?
Yes, please make it full screen, please.
Sure. All right.
Yeah, I can see your full screen.
Okay. All right. So not to further ado, let me start. So for the one who don't know me, my name is Thanet Jantadet. I'm based in Thailand. My position is the Regional Application Support Specialist. So the topics today, we will go through the comprehensive solution for the Oncology NGS, including the Chi-seq targeted DNA, RNA, and the multimodal DNA and RNA. And actually, we just launched the long lead kit, so we will add it for you in the last part as well. So let's see the agenda today. So we separate into the two parts. So for the part one is the overview. We will go through the cancer incident in Vietnam, molecular genetic testing for the current, and sequencer platform and the Chi-seq compatibility. Then we will go to the second part later. Let's go to the first one. So as you can see, I got this number from the WHO 2022. It's quite a bit old, but that is the update one. I can search it for now. Anyway, as you can see that the number of the new case now is almost up to the 200,000. Number of the pass away people that caused by the cancer is around 100,000 as well. And if we count 5 years, it's almost 500,000 that related to the cancer effect to the life for the life quality for the Vietnamese people. So, but anyway, if we think in the positive way, this number maybe it's happened for 5 to 10 years already, but now Vietnam have developed and upgrade about the instrument and technique to screening the cancer more and more advanced. So that's why you can detect more and more number of the cancer incident and the number of the people that pass away by the cancer. So anyway, this cancer, we believe that finally since we can detect the early stage of the cancer. So now with the power of the precision medicine, we expect that we can treat the cancer people with the precision medicine as well. And QIAGEN have that solution. We will go through it today. So if we separate by gender, for the male in Vietnam, the number one cancer is the liver, lung, and the stomach. For the female, is the breast, lung, and the colorectum. For the both sex, the number one is the breast cancer, and the liver, which come from the man. Yeah. So now, I try to find another information and found that the risk key factor actually is come from the first of it is come from the infectious agent. HBV is the cause of the liver cancer. HPV is the cause of the cervical cancer. Tobacco, definitely lung, throat, and another cancer that related to the respiratory tract. Alcoholic consumption related to the liver, colorectal, and another digestive tract. Yeah, and environment as well. PM 2.5 definitely a lot now around the Southeast Asia is the incident for the lung cancer. By the way, I know that Vietnam government try to set up a lot of campaigns to reduce this kind of the key risk factor. For example, for the infectious agent, you have the vaccinate program to reduce the HBV case that related to the liver cancer. You also have the tobacco control. And importantly, you also collaborate with the international partner, which the WHO, that they concern and set up about the national cancer control strategy via genetic testing. So the take-home message here is, if we know what drives the tumor at the genetics level, how might that change the way we diagnose, treat, or even prevent the cancer? So when we talk about the genetics level, the current protocol is the PCR. Some lab use the microarray as a gold standard. Some lab use the Sanger, or even some lab now use the NGS or the next generation sequencing already. Each method have the pros and cons. I will not go through it in detail, but just focus only the NGS because that is the technology that we will go through it today. Yeah. Next generation sequencing, even though it's the high throughput, when we talk about the high throughput, meaning that you can do the sequencing, you can get the sequencing data for the multiple sample at the same time of the running. However, to get that one is not easy because it's very expensive technology. You need a very high budget. It takes turnaround time quite a lot. Maybe from end-to-end process is around 5 days. Comparing with the different method, maybe you can get in 1 day. Yeah. However, you also need the skillful technician as well that know how to do the library preparation and the bioinformaticians to analyze the challenge data that come from the sequencer. However, that solution, that issue, we have the solution from QIAGEN today already that make your life a bit easy for the library preparation in the wet lab part. Also, for the software during the secondary and tertiary analysis part, which will be presented by my colleague in the next presentation, Dr. Shiva. Yeah. So the generation of the sequencing, if we separate now, we can separate into the 3 generations. First generation is the Sanger sequencing. So some of you may know this machine already. Yeah. Second generation is still can use until now, which is the short read. And the third generation is emerged in the market more and more this year until last year already actually. So the second year and the second generation and the third generation, this is the up to date sequencing technology that's still available, and QIAGEN have the solution for both second generation and the third generation already in terms of the wet lab and analysis part. So if we go to the sequencer platform, so as I told you that we have the two separate type of the sequencer. On your left is the long read one. As you can see that PacBio have and the Oxford Nanopore is the main company that related to the long read. Meanwhile, for the short read that you may familiar with the sequencer platform is the Illumina MGI S5 from the Thermo Fisher, GenMine. PacBio also established the short read as well recently, which is the Onso, yeah. And you may heard about the Element and the Singular and the Ultima, which three of these one used in the Western and the EMEA country. So now let's see for the sample to insight or the workflow that QIAGEN already have the solution. So the first presenter also tell you about the sample preparation already. That is the first part that you have to do by extraction from the raw sample, even though it's the fresh sample, blood, serum, or even the cell-free DNA, urine, saliva, we have the solution already. Even you want to do by the manual or by the automate, we have the solution already. So I will not go through it on this. Once you have the sample preparation, we need to do the sample QC. So the sample QC, normally what you need to do is check the concentration by the qubit or I recommend use the fluorescent method, and you check the purity by the QIAexpert, and then you do the library preparation depend on the panel that you interest, and once you finish the library preparation, you have to do the library QC. Normally, what we need to check is the concentration and the size. So QIAGEN have the QIAxcel to allow you to check the library size and the primer dimer contamination as well. All right. And then we have to go to the primary analysis by the sequencer, which this is only step that QIAGEN don't have. QIAGEN normally we ever have the sequencer, but it's obsolete already. That one is the gene reader. So that's why we try to develop the library preparation kit to make it compatible to all sequencer type which available in the market. Even though it's the short read or the long read, we can run in all the platform now. Yeah. So that's why for the NGS kit, QIAGEN use the U in front of the UNGS kit, which come from the universal, meaning that our library prep kit can run in compatible with the all type of the sequencing platform by now. Yeah. Once you finish the library library sequencing, you will get the raw file FASTQ, go through the secondary analysis by the software that QIAGEN have, CLC analysis. You got the variant calling profile in the VCF format. Then you go to the last part, tertiary analysis, for annotate the data until get the clinical report. So the doctor will know which precision medicine that available in Vietnam and should treat to the particular cancer patient. So this is overall from end to end that QIAGEN already have the solution. Yeah. So just want to give you a brief idea on the first paragraph here. This is the name of the main QIAseq product. When you heard about the QIAseq, it means the name of the library preparation product. Yeah. And on your right here is the name of the sequencer. The green one meaning that it's compatible and we already validate on that machine. Yeah. For the yellow one is the one that compatible, but we not yet validate. Meanwhile, the red one is the one that not compatible. So as you can see, the red one is related to the long read sequencer, which most of the kit is not available, but just only one, which we just launched this month, I would say, which is the QIAseq X-Type long read panel. We will go through it in the last part, yeah. And another platform that got a lot of the red light is the Ion Torrent, which is the single read. So the reason that it's not compatible because of the most of the panel we have is quite huge. So that's why the capacity of the chips for the Ion Torrent maybe it's not enough, but for some application, for example, especially for the onco solution here, fusion for the RNA fusion detection and the microbial is available for us to detect. Okay. So
It's the activity.
that's it. Finished for the first part already. Now we go to the second part, which related to the end-to-end workflow analysis guideline. And the new panel that we have is the CGP panel in the long read solution. All right. So let's go to this page again about the sample preparation. Just want you to get some idea that for the extraction part, we have the solution for the DNA-seq application, cell-free DNA-seq application, RNA-seq application, or even the multimodal, which allow you to prepare the DNA and RNA library simultaneously. So all sample type for each application we have available here already, even though it's the standard DNA from blood, FFPE, or even the cell-free DNA. We have all for the each type of the application already. Okay, so if you want to get more detail about the each extraction kit, you can text to me later, or you can contact to the marketing team or even the first presenter who is the expert for the extraction kit. So now we go to the library preparation part. So I designed to separate by the cancer analysis guideline. Normally in Thailand, we separate into the 3 parts, which is the low risk of the individual person, intermediate risk, and the high risk of the cancer patient, I would say. So let's start with the low risk. Low risk is the individual with the family which has the cancer. So when we talk about the history of the family who got the cancer, what they want to detect is related to the germline, yeah, not the somatic, but the germline itself. So the risk reduced. So it depends on whether after they detect by the next generation sequencing, they can see something or not, or some signal or not. So we have the KAISIC Comprehensive Hereditary Cancer Research Panel, which combine several types of the cancer and have the gene list around more than 300 gene. Yeah. So it depends whether we can detect any cancer or not. If not, we still able to explain to the individual person that you can come to the hospital and get the body checkup every year based on the age, based on the gender and the sex, which I will show you later for the criteria. Yeah. So before that, let's go to the gene list that I just explained you for the low risk individual person. So the name of the kit is here, Hereditary Cancer Research Panel. Is composed of the 317 gene, can detect 12 cancer, and these main 12 cancer also have the 124 cancer subtype. Yeah. If you want to know more about what type of the cancer and which gene related to what cancer, you can go to this link. Okay. So where exactly is located on the our panel list? So is here, Comprehensive Hereditary Cancer Research Panel that I just suggest you for the screening, the low risk purpose. You can run in the MySeq using the correct depth around 500x. You use the V3 flow cell type, you can run up to 7 samples. Yeah. But we also have the several type of the panel which allow you to detect the germline. Yeah. So later on, you can come to this slide and check whether it's catch your interest or not. The good thing is that, let's say that you interest about the colorectal cancer panel for the germline detection, and you want to modify it from this catalog panel by adding or delete some gene. You can do that. We have the customized panel. So all of the kit here is the customizable. All right. So this is just some guideline in case that the customer join or applying to the low risk program that's set up by the hospital and they cannot detect anything. So you're still able to suggest them to do the body check separate by the gender and the age, range of age. So that is the way that the hospital still can follow up with the customer. So in terms of the intermediate risk, so normally we suggest with the one that probably have the healthy, didn't see any signal, or they feel healthy, but once detect the blood, they can detect some things to indicate that, hey, you have a low risk, you have the some risk of the particular cancer. So this one is related to the somatic. It's not the germline anymore. Yeah. So we have the solution named KISEEK, human actionable solid tumor. When you heard about the actionable, meaning that the biomarker or the gene list in this panel is is known, is the one that we know available clinical action, meaning that we know the precision medicine list for this kind of the gene list in this actionable panel already. Okay, so that's why for the individual people that join this program based on the indicator from the blood, they can go up to the precision medicine. Yeah. So if you're curious that what indicator I mentioned, so here I list for you already. This is the list of the indicator. I will not go through it in detail, but from time to time and many publication now, they know that several indicator is related to the specific cancer type as well. So we detect from this for the intermediation program. Yeah. So now when we talk about the gene list for the actionable solid tumors, this panel composed of the 30 gene list, which composed of the actionable gene list related to the several cancer as listed here. Anyway, as you can see that for the colorectal, there are just only the 5 genes. And for the another type of the cancer, they also have the different type of the gene, but it's not that much because the purpose is just only for the screening. Yeah, whether you have, based on the indicator that we can detect, whether you have any risk of the actionable gene that appear in this panel or not. All right. So where this kit locate is located in this page. Okay, it's the different chemistry we call the V3. Yeah. So as you can see that apart from the actionable solid tumors, we also have the different type of the panel based on this chemistry, including the cancer related and the non-cancer related. The non-cancer related that I mentioned is for example, the mitochondrial panel. We also have the pharmacogenomics. We also have. Yeah. And this one, I guide you the sample per run in the MySeq, NextSeq for the somatic that fix the variant of frequency at the 5% and the depth at the 7,200. You can run up to this number. Yeah. Based on the different type of the sequencer and the flow cell use. Okay. Anyway, if your lab have the different type of the sequencer, could be the MGI or Element or the PacBio, also, and you want me to calculate for the sample multiplexing, I'm happy to help. Just text me, yeah, or send me the message later. So now we go to the high risk. Who gonna be join the high risk is the one that individual that already diagnosed with the cancer. They know what cancer they got already. So definitely it's go to the somatic. And we have so many specific onco cancer panel. For example, if they know that they got the lung cancer, so we have specific lung cancer. This one will be better than the actionable because actionable, we also have the lung cancer, but it may have the actionable gene just only the 5 to 10 genes, not more than that. But if you have the specific onco panel here, you have the gene, I think more than 100 genes that related to the lung cancer, which allow you to get the greater number of the precision medicine choice, whether so it will be more specific and more choice for the doctor to consider whether which one should treat to the patient. Okay, so this is some just brief idea for the low risk, intermediate risk, and high risk, which kit should fit which application. Okay, and where it fit into the portfolio. So this is under the different chemistry is V4. With the power of the V4 chemistry, you can finish the library preparation in half day. Because if you know how to do the library preparation, the tedious part is the cleanup step. So we not use the cleanup step with the bead cleanup anymore. With the power of V4, we use the enzymatic cleanup. So we engineer the enzyme to recognize only the short fragment, which normally is generated from the artifact and the primer dimer. So that engineer enzyme will digest that one, and the output you can go to the next step. So that's why the turnaround time is very, very short. Once the customer change through the V3 to V4, they don't want to go back to the V3 anymore because the turnaround time for the V4 is pretty much shorter comparing with the V3. And you can prep and start run the sequencing in the same day. Yeah. So what panel we have? The one that I just showed you in the previous slide is here, somatic hotspot. Yeah. Breast cancer, colorectal, myeloid, brain, and the lung. However, all of these cancer, we also have for the somatic full coding as well. But the full different thing for the full coding and the hotspot is that the full coding will cover for the whole exon. Hotspot will cover only the particular nucleotide that we know already that is the tend to be the pathogenic mutation if there is some change on that hotspot. Yeah. So it depends. If you want to detect the hotspot, not the full coding, definitely you can run more sample per run in the same flow cell. For example, breast cancer, for the somatic hotspot, you can run in MySeq V3 up to 11. But the same panel with the full coding, you can run just only 3. Because if you want the full coding, you need more read per sample in the equal flow cell, yeah, comparing with the hotspot. So this is some idea that I just want to give you for the targeted DNA. Anyway, if you think about something bigger, something huge, more than 500 gene lists can detect anything for both DNA biomarker and RNA biomarker, we now have the solution. The name of the kit is the QIAGEN multimodal DNA/RNA library prep kit. So this is the backbone chemistry, and they use enrich by the probe hybridization, and the probe that we have now, the huge one called the CGP panel. CGP comes from the comprehensive genomic profiling. How the workflow look like? All right, please allow me to skip this one because I afraid that it will spend too much time for you. So what is the CGP composed of? So it can detect the DNA biomarker up to the 724 oncogenes and tumor suppressor gene as well, meaning that the TMB and the MSI we also can cover. Copy number variation and loss of the heterogeneity we also yeah. DNA translocation, we also can detect. So that is the DNA part for all that we can detect. I believe that this kit is the biggest one for the current library prep kit that available in the market. Yeah. We also can detect the RNA marker. Normally, when we talk about the RNA marker, it will including the expression and the fusion. Fusion that we can detect is including the known and novel fusion as well. So we have another 234 genes. Some genes here in the RNA is overlap with the DNA, but not all of them is overlap. Yeah. If you're interested about the gene list for the CGP panel, what it composed of, you can send a message to me. I'm happy to share. Yeah. Cancer type that we can detect is available in this list already. Okay. So now let's see the workflow, how the multimodal allow you to detect both DNA and RNA. Yeah. Start with input material. You can add the total nucleic acid, which composed of the DNA and RNA, which the last presenter also mentioned about the all prep kit, which allow you to extract the DNA and RNA from the same sample. But that DNA and RNA will available in the separate tube. Okay, so it allow you to detect the concentration and do the QC for the DNA part, and the RNA part as well. Yeah, so that is the good thing of the all prep kit. Yeah, so let's go very brief for the workflow. In the beginning, you have to insert the DNA and RNA here, okay, which you can do the concentration check and the QC check as well, as I told you before. So I count it as a total nucleic acid, yeah? So we start with the fragmentation for the DNA and the fragmentation for the RNA by the heating. DNA will do the end polishing by the do it blunt end and A adding. And then all RNA that fragmented, we will do the A adding, poly A adding, yeah. And for the DNA now, we do the adapter ligation. And from this step, you will have the complete DNA library already and the RNA that not convert to the cDNA yet. So we split this one into two parts, for the DNA part and for the RNA part, okay? For the DNA part, is ready to go to the whole genome sequencing. If you want to get the whole genome sequencing data, you can use the library prep from this step to run sequencing directly, all right? But for the RNA here, now there is the DNA and the RNA, right? So we will treat with the DNase to eliminate the DNA library and remaining only the RNA, and then do the reverse transcription to change only the RNA to be cDNA. And then that cDNA will do the indexing and is ready to go to the whole transcriptome sequencing. If you're interested to generate the data for the whole transcriptome sequencing, you can go to the next generation sequencing. Anyway, if you want to narrow down, interesting just only the gene list in the CGP panel that I just told you, we can do the hybrid capture at this step. That probe, we can do the customization as well. And also is compatible with the third-party probe for both DNA and RNA. DNA, the third party that I mentioned is available here from IDT, Kappa, Agilent, and Twist Biosign. You can use with them to capture the target of interest by this step for the DNA and at this step for the RNA-seq as well. Or you want to combine the CGP panel with the third-party panel in the same time to capture for both target gene in the CGP panel and the third-party panel also can do it. All right. So if you're curious that you need to if you interest only the DNA part, but you don't interest about the RNA part, you need to purchase two of them or not? The answer is no. You have a choice. You can purchase only the DNA, the backbone chemistry, just only the DNA, or backbone chemistry, only the RNA. As shown here, this is the QIAGEN page. As you can see here, there is the CGP DNA, CGP RNA available for you. So it's not forced you that you need to purchase the DNA and RNA, all right? So it's quite very, very flexible here. So by compare with the competitor, we can detect more. Now we are the best one that can detect more list of the DNA gene and the RNA gene at the same time. Customizable is available. You can finish the library prep in 1 or 2 days. It's available for the workflow for the FFPE and the cell-free DNA. Input DNA is the 40 nanogram. And the best one is that we guarantee that the price per sample is cheaper than the different company for sure. All right. So now is the long read that we just launched last week. So, all right, I will speed up. We have the two panel for the long read now, which compatible with the PacBio long read platform and also the nanopore. So if you have this name of the sequencer platform available in your lab, you can run these two kits, which is the hereditary cancer research panel and the HLA typing. Let's go to the, okay, I will skip this one. Let's go to the hereditary cancer panel first. So this is the list of the gene for the hereditary cancer for the long read. We have up to the 95 gene which related to the structural variant. That's one you can detect, and we do validate in PacBio already. It requires just only the 0.4 million, and you get the 40x coverage. If you go up to the 1 million per sample, you will have the 100x coverage. What type of the structural variant you can detect? Long insertion, long deletion, inversion, interspersed duplication, translocation, and the tandem duplicate. All of them we can detect by using this long read kit. All right, now we go to the HLA. HLA, we cover for both HLA class 1 and HLA class 2 is cover up to the 18 full length of the HLA gene, which cover not only the coding part, but also the intron and the UTR as well. So for the one that doing the application about the HLA, I believe that you will be happy on this. Okay, and the application that use is the transplantation, immunogenetics, and the disease associated study. All right. So this is my last slide. So the kit that we just go through it today is just some part of the targeted DNA, yeah, and the targeted RNA is fusion here, and also the multimodal here, okay. But apart from this, we also have much more kit which related to the microbiome. For example, if you want to do the whole genome sequencing or the bacteria virus, we also have the solution. You want to study the 16S expression profiling for the microbiome, we also have the solution. Epigenetics for the methylation and ChIP-seq, we also have the solution as well. So if you need more information or any additional data of the sequencing, do not hesitate to contact me or my colleague, Mr. Thang. I provide you the mail here. All question is welcome here. Thank you.
Thanks a lot, Thanas. Very nice information. Very nice talk. I have a question. So the slide number 25. So I see you use a rhymer in the header of the timetable. The slide number 25.
25. Okay, let me go through it. This one?
Yes.
All right.
So
your
you
question
said
again?
that the column number 4, the total number of primers. So it means that you use the PCR amplified, not the probe you
Correct.
target from.
Yes, for the targeted DNA, for your information, targeted DNA, we have the V4 chemistry and we have the V3 chemistry. So these both chemistry, we use the amplification method. So we have the specific primer to amplify the gene of interest. But normally, if you know, I'm not sure whether you got this information or not. Normally, the way QIAGEN amplify, we use the single primer extension. Each specific target that we want to detect, you need just only one specific primer. Because another site, we will use the universal primer. So this one allow you to reduce the bias of the primer dimer and also provide you the more specific during the primer design. Yeah.
It means that, like for example, you have 633 primer to
Yes,
amplify.
correct.
And
Correct.
it means that you run the multiplex PCR in one reaction chip only.
Correct.
Oh my God. It's a
Yeah.
lot to optimize
Yes, it's a lot. Or even the comprehensive. As you can see, now
Yeah.
we use the AI to help us to do the calculation and design for the primer to detect the specific region. Because definitely if you do it manually, it's very tedious. If you have the gene, like it's impossible, I would say. Yeah, so that's why we have the supercomputer to calculate for the to help us to design the specific primer that can do multiplexing in the same time.
So can you give me the name of the software or the paper that provide the open source software to design this primer? Can we have this software?
Well, it's the for the software, I afraid that it will be confidential, but you can use that one to design the target of the if you have the target of gene list and you want to use these tools or this software is available in the gene globe. Yeah. Or you
In
can
the gene globe, where is
yeah.
it? What is the name?
Gene Globe is the QIAGEN website. If you go to the please allow me to you still can see my screen, right?
Yes, I can see your screen.
Yeah. You just type the Gene Globe, yeah. My internet is slow a bit. Sorry about that. If it's still slow, later on I can send you the link. Okay.
Okay,
And
later
yeah,
on,
yeah.
later on, you can
And then you can go to the custom product. So here, if you scroll down, just a moment, yeah.
I cannot see your screen now. I just see the slide at the moment.
Oh, just a moment, just a
It's
moment.
okay. It's okay. Later, you can show me the YouTube to now I
Sure.
can see it. Yeah, now I can see.
Okay, so this is under the gene globe, yeah. You can search gene globe, and I believe that it's come to the first link in the Google, and then you go to the custom product here. Okay, you scroll down a bit. You choose the next generation sequencing here. You select the analysis type, whether you want to generate the specific primer for the DNA, RNA, or you want to do the multimodal. Multimodal is mean that you want to detect the DNA and RNA at the same time.
Yeah.
For these three is the specific primer based, okay? But the microbial here is the hybrid capture based. Okay, so let's see for the DNA, and then you go through it, whether you want to do the custom human, targeted DNA Pro, X-Hype custom microbial, and much more data. So let's say that you want to go to the targeted DNA Pro, which allow you to detect the single nucleotide variation, copy number variation, and the indel. Yeah, all the biomarker in the DNA you can do it. Then you click Open Design. Then it will go through the page that allow you to put the name of the gene list. All right. So you just put the name of the gene list there, which could be the yeah, you have to type something here, and then if you go to the next, I think I will not show you in the next step because it will take time. Then if you click next here, in the next page, it will allow you to put the name of the gene list that you want to design. And also it allow you to pinpoint whether you want to sequence the whole part for the overall exon, the hotspot, or including the UTR as well. It have that option for you as well. Yeah, so it's quite flexible and comprehensive.
Yeah. So it's free of charge to do the analysis like this one to design the
Correct. Yes, for design, in silico design is free of charge. And if you want to save your time and if you don't mind, you can share that gene list to me. I can do it for you because normally I will not use the gene globe, but I will contact to the enterprise team in the US. So you can got the base file in one day normally.
Okay,
Yeah.
thanks a lot. And then my second question, so in the lab panel about using a long read to evaluate some kind of gene panel, do we have any IVD for the solution, the
No.
IVD solution?
No. Normally the NGS kit, all the NGS kit from QIAGEN now is the RUO. We have the reason behind that. As you know that the NGS is developed even day by day these days. All month you will get the new kit emerging from the different company. And let's say that this one normally is reflect by the update of the publication and the guideline from the different type of the data source, including the WHO as well. So to wait until we develop the IVD kit, I don't think it will be on time with the I mean it will be uh, immediately used for the patient that really need to diagnose. So normally, I understand that this law is vary country to country. Some country they fix the rule that you have to use the IVD machine and the IVD product to analyze for the oncology until get the precision medicine list to treat the patient. But in Thailand, normally they allow to use the IVD machine, but for the kit, you still can use the RUO. But you need the geneticist to sign, or the doctor who know about the genetic counselor. I mean, yeah, to sign whether they approve this data from our software or not. Yeah, because
Okay.
our software normally it just provide you the list based on the very huge data that we available in the QIAGEN knowledge base. Anyway, some genetic counselor, they also want to screen another time as well before they will choose the precision medicine list to treat with the particular patient. Yeah.
Okay. So we have one question from Dr. Huyền. She asking for the hereditary cancer panel using a long read sequencing. Have you validate on a nanopore system?
Yes.
And then the second question, what is the cost compared with the short read sequencing? Thank
Okay.
you.
Interesting. So the first question, have we validated in the nanopore, right? Sorry.
Yes, the nanopore.
Okay, the answer is yes, we validate in the nanopore already. But since this slide, I just got it, I think last 2 days, so I just add it. It's still warm, if you can feel.
Yeah.
So
It's very hot now, very hot. Still
yeah,
hot.
it's very hot. So I have now just only the PacBio, but the R&D didn't send me the result for the nanopore yet. But definitely, if you can see this slide that I just skipped, sorry about that. Normally the nanopore here it can run up to the 2 billion base pair. I mean in the length unit. But it will compatible with the HLA and the hereditary cancer. Yeah. But anyway, you still need to adapt the library once you finish preparing by the QIAGEN product. So what you need to modify it is by the enpolishing and a adding here. Because you have to do the enpolishing and the A adding at the 3' end for the both side to allow ligate with the index from the nanopore. Because normally the index from the nanopore will compose of the protein, which will recognize the pore protein is the mobile protein, if I remember correctly. So this one will bring the fragment, whatever that we generate the library, go through the pore and push the single strand of the pore in through it. So it will fluctuate for the ion that is normally it will go through the pore as well. So with this fluctuate pattern to the pore, it will go to the signal like this, and then the machine will read it for you whether this pattern is gonna be the A, T, C, or G, something like that. So first answer is yes, it's compatible with the nanopore. However, the price, I don't have that information in my hand now. I just have only for the just only the number of the maximum sample per run, just that on the nanopore. Yeah.
Okay.
Yeah.
Thanks a lot. Have any question? Mấy bạn có thể hỏi tiếng Việt rồi tụi tôi, nếu mà tôi không dịch được thì tôi nhờ mấy anh chị khác dịch giùm. Đó, đừng có ngại mọi người, có ai hỏi tiếng Việt không? Hỏi tiếng Việt cũng được mọi người. Ở đây mình không phải thi tiếng Anh. Đây mình muốn học cái này chứ không phải mình thi tiếng Anh muốn biết. Đó, bởi vì không cần phải giỏi tiếng Anh đâu mọi người, cần. Đó, tôi muốn tổ chức cái lớp để mọi người học được, chứ không phải vô đây để mình khoe tiếng Anh của mình, không có nhu cầu đó. Có ai hỏi nữa không mọi người? So my last question concerning about the 3 panel you show about the low risk, intermediate risk, and then high risk of cancer. And I did not see, or I missed anything that about the, I didn't see about the CT, CFP9 for liquid biopsy testing. Over here, always using the FFPE. For a low risk, maybe we can use the blood sample, but
Okay.
not the CT, CFP9. For intermediate risk or the high risk, what kind of sample input we using?
It depends on the cancer type. For example, if that cancer is related to the lung, which very difficult to biopsy, you can use the cell-free DNA. Yeah. But definitely the cell-free DNA, you really need the coverage depth very, very high, because especially the early stage, that the apoptosis for the cancer cell is the rate will be very low. So if you want to detect from the blood, is would have the false negative. So we actually have another kit we call the cell-free DNA Ultra. If you don't mind, please allow me to go through that slide for a while.
Yes.
Yeah.
Yes, please.
Okay. Just a moment. Yeah. All right, here you go. So, first of all, it depends on the cancer type, whether it can biopsy or not. If yes, so it's pretty easy. You can do the FFPE, yeah, which 0.5 variant of frequency is more than enough. And we have that solution. And the targeted DNA, you can detect for we have solution for the cell-free and for the FFPE as well. However, for the early stage of the detection or even the follow-up after you treat with the precision medicine already, you definitely want to know that the last single cell of the cancer is gone yet, else it will be elapsed or it will be come back. So we have another kit is called the cell-free DNA Ultra. This one some lab even apply for the MRD, is the measurable I forgot the name already. Yeah, it's the MRD. Yes. So that MRD we definitely need the coverage depth up to the 48,000 and can detect the variant of frequency down to the 0.1 here. Okay, so we have this kit and it's available for the cell-free. Okay, especially for the in the scenario that it's very difficult for the biopsy and you really want to detect in the early stage. But anyway, this ultra depth, you also need the very high throughput of the sequencer. So for the Illumina, you have to use the NextSeq. 1,000 is fine, 2,000 is the best. But 2,000 you have to go with the P3 for flow cell. If you ask me whether MGI high throughput, for example, the MGI 2000, MGI 50, I believe that it also can run, but the number of the sample per run may be down to the just only single number, could be around 6 to 8 or something like that. Yeah.
Yeah, thanks a lot for your information.
No
So,
problem.
có ai hỏi gì nữa không mấy bạn? Không hỏi thì mình qua phần mới ha, phần cuối cùng, phân tích cái kết quả, được không mọi người ha? Thanks a lot, Thanas. So now we going to the last presentation.
My pleasure anytime. And just want to emphasize again, if you want, if you have any question or would like to know about the detail about the kit, and also my colleague just sent me that if you want to know about the price for the long read kit, particularly for the hereditary cancer panel, the local cell team, happy to share with you on that. Okay, so we will get back to you later on that. But for the technical part, please feel free anytime to contact me or my colleague here. I put it in the last slide. Thank you.
Thank you. So now, Siva, on your.
Thank you, Thanat, and thank you, everyone. I think we are on the right track. Just allow me to share the screen.
Yeah.
Okay. Okay. So I think we had the discussion till now for discussing like how do we start with the starting material, the tissue or the sample, what we start with. The next, how to use the panel, build the panel, and how QIAGEN helps you to build those panels. And then the final thing, how does this analysis, because this NGS analysis itself is very complex and requires a lot of inputs. So that we have a fear that the analysis of data becomes very cumbersome. You need to have coding language or bioinformatics knowledge. So really, how does QIAGEN team is trying to help you out? So we, the sister department within QIAGEN, we are working especially as a digital partner for QIAGEN to provide the analysis. So this is what today I will be walking you through. Okay. So ultimately, when we started today's session, the first two speakers also, the ultimate goal of this clinical scenario or clinical doctors like you who wants to have is a report. That report which clarifies you what is the mutations or molecular alterations, whether it is TMB, fusions, CNV, or single nucleotide mutations. What are their classification? Are they really pathogenic to a particular disease? What is the phenotypes it is given? What does the guidelines like NCCN, AMP, American Molecular Pathology, what do they say about it? What the ClinVar says about it? Are there any clinical trials which are ongoing? Is there an approved drug available for that? Is there a combination of drugs available for that? That is the relevant information which you want to treat a patient. So that's the ultimate goal. From the sample you gave to us, then we want to develop a report which is much more easier. You will be able to create a valid report and develop and report it. Okay, so that is where you have one of the best hands which provides you the references, the evidences, and all the interpretation. Okay, so this is where we are going to help you out. So as I was telling you, the complexity of NGS analysis, even your lab has tested thousands of samples, but still sometimes you find a new mutation or a new novel variants. There is a chance possibility. So you may not be sure what does Cosmic database says about it. Did any guidelines like European or US FDA, they have changed anything? Is there a new clinical trial for this mutation which is going for this, has been approved or not? So all these questions are there. So especially the complexity of that, what I showed you in previous slide. Okay, so the NGS data interpretation and reporting, that's where the context you need. Okay, so you have a lot of panels and a lot of things are going through. You have thousands of variants. You need to filter them down. You cannot look into all the thousands going in detail. So you need a variant which helps you to report it. Okay, so this is where we want some software or the support which helps you to confidently, the most important thing, confidently call the assessment, because this is going for a patient treatment. Okay, automated variant analysis, and it has to be evidence-backed, the results which you ready, and also easy to generate a clinical ready reports which helps you to do this. So this is where we have these tools which are helping you out. So the first thing is you have the FASTQ generated from the instrument, irrespective of what instrument you generate. We have this CLC Genomics workbench, which can do your data analysis, like taking the FASTQ file and generating a VCF file. So that identifies the variant. Okay, so that is a tool which does. The second is you have taken the VCF file. So that VCF file has all the variants informations. What are the mutations? What are their configurations? Everything is there. So now you have a cancer type. That patient had a cancer type. So again, at the disease, what does our knowledge base says, and what is the guidelines especially says that, and that it brings all the annotation to that, and it helps you to report that in a PDF format. Using a genetist and oncologist, you can sign out that report also. Okay, so this is where we are really want to help all the researchers who are in this field, or the clinical CROs, or the clinicians as such, who looks for improvements like process improvements, better knowledge, and improved technology from both the perspectives, somatic and germline, having it and running faster. We are happy to help you out. Okay. So coming to the first tool, like as soon as the panels you run, okay, whether it may be the QIAGEN panels which Sanat mentioned right now, or any other panels you run, you have the data generated in the FASTQ or some format. So we have the tool called as CLC Genomics Workbench, which has been there for almost 25 years till now. It's one of the oldest secondary analysis data which we have. Okay. And the beauty of this tool is it can run from Sanger data also. It can run IronTorrent, Illumina, ONT, PacBio, BGI, MGI, and Element, many other tools. Okay. And it does also run on Windows, Mac, and Linux. You don't have to only use coding programs. It's very user-friendly, very simple. It can do your variant detection, calling all these things in a very simpler manner. Okay, so they have been a lot of different analysis types it can do. It's not only applied to this one, it can do multiple other things. Like even if you are interested in research side and you want to do single cell analysis or targeted transcriptomics, you have this one. Okay, so that's adding to that. So today you have only 100 samples. I want to run it on desktop, but your simple panels becomes more complex, like the TSO 500 or the Cancer Comprehensive Genomics panel, what my colleague presented before. And so when you increase more number of samples or more data, you will require a server, or you may go to cloud also. So the same workflow can be installed and run in the cloud module or in the server. So that's what the viability of these tools we provide. Okay. So then, how you see, I explained so much. If you ask me a question, Shiv, how this is going to help me out? See, it's very user-friendly, where within 5 to 6 easy steps, you just upload your FASTQ, give the bed file, and tell what is the allele frequency you want, and all the annotations files you provide. It provides you the results analyzed with the standard tools, where you will have a IGV like browser view, where you can have a mutation. And let me show you this, where you can see this. When I select a mutation, the list of table which has the mutations and annotations, everything, I can select the mutation. I can look how this is mapped to reference. How are my reads? How much is forward and reverse? And I can go through each of these reads, and whether they are in the CDS coding region, are they leading to amino acid change, and whether there is a mutation which is relevant to that. So I can exactly validate this through. Okay. And I can also check if I had my adapters and primers list, I can check whether this is the region which is overlapping with the primers or the bed file. Okay. So in this way, it's a very nice graphical user interface. You know, it's not just the coding where you don't know what's happening inside. It's a visual treat which will help you to definitely analyze and how it goes. Okay. So all these features usually like user-friendly interface, very like it just guides you through how to do the analysis. Like I showed you in the last slide, you can visualize and look at the mutation and the workflows. There is end-to-end workflows which are helpful for automated processing, and you can share with the colleagues also. And many of this, like almost many of this, the panels from QIAGEN are well supported with CLC workflows. Okay. And it is ISO certified, and a lot of GMP labs also, big labs are also using it because it has audit log as well as fully documented, and everything is well supported. Okay. So with this, now we had a FASTQ and we generated the VCF file here. So now the thing is, how do you translate that information in the VCF file into a report which is benefit to a clinician who is trying to help a patient? That's the ultimate goal, what we discussed. So now, for that only, we have a tool called as QIAGEN Clinical Insight. Okay. So feel free to reach out any questions. I'll be going here. So as we discussed, this is the second part. So where QCI takes the VCF file and tries to generate a report. So this is where the biggest thing, like today, a lot of people say we have everything, like I have artificial intelligence or I can manually curate it. But we have a team which has been doing this for 20 years, more than 25 years. And we have experts who interpret, experts like clinicians, oncologists, researchers like you, and also the geneticists. So who all sit through and curate the content, okay? And we also have AI and language programming so that it prioritizes some of the variants or the data from the web pages and provides us. And also databases like Cosmic, ClinVar, HGMD, OMEM, and I think almost 40 to 50 databases, we have already integrated it. So if you ask me, like Shiv did last week, there was a clinical trial which was rejected. So then we would have done that in the knowledge base. You don't have to worry about it. So the any like US FDA has approved a new drug last 2 weeks back, so that I can share you it has been added to this one. So we are up to date content, and we are looking for the consistency and the accuracy. This is where we are a trusted content with HIPAA, ISO certified, and also recently we are from the European country perspectives. We have also got IVDR certified, okay? And all these things have been well recorded in the documentation. So this is where, like, you know, what I was telling you, the key benefits is you don't have to look at 10 different places. Like I am going to I am not going to clinicaltrials.gov where something is going on, okay? I can do all this analysis by myself, and I can do this analysis with use of QCI, okay, which provides me from each and every patient's perspective what is the molecular function, what is the incidence and guidelines, how it works, okay. So then, knowing this is the knowledge base, what is sitting behind. How easy or how difficult is this one? So it's a very user-friendly tool. Again, we try to make it any simple person, even including clinicians, who wants to upload their samples and they visualize the information. It's very easy to follow the steps. And it is through easy 3 steps. Where first step, you upload the VCF file, provide all the patient information, relevant ones. And as within the knowledge base, already there, for each of your variant, it will tell you what is the criteria we provide. And that you need to review the evidences we provide and assess the classification, which computed classification is provided by the software also. You can look at the evidences and agree or disagree on that. So it's in this way, it's an open tool, which always you can do. Also, we have an option, we call it as QCI1, where you can submit it for our expert interpretation also, okay? So we have a team which can help you out for the interpretation also, okay? And this is where the list of variants, you will find it, and you can look at like TMB, MSI, HRD score, all these things you can look at, and it provides you with a pathogenicity and also actionability as per the AMP and ACMG criteria with all the relevant informations, where you have AMP, CAP, and ASCO guidelines and pathogenicity by ACMG, and then the reportability. And that's what I was mentioning. You always have the open end where if you disagree with the evidences we have provided, or you want to add more rationale to this, it's always easily doable. Okay. And the very important thing is, if you work with larger panels, okay, then more number of variants will be there. So that's where we have settings where you can work out some filter settings of confidence or common variants from the normal population. You can filter out and reduce, work on less number of variants annotation, okay? And also, as you can see, like I am interested to look at only the CNVs or the tier 1A actionability ones. So you can visualize on those by setting on this one. So this settings also helps you to look at only the relevant informations. And also in the variant details page, it gives you what is that software is telling you about the competent classification, what is the effect of mutation, whether it is leading to a gain, means it is in cancer perspective oncogenic role or a tumor suppressor role. What is the impact of the mutation? What is the allele frequency from the reads you got? And you know, the beauty of this is it has cosmic database also, which tells you what is the variant frequency, and also the in silico tools we have, which predicts the whether having this mutation leads to a deleterious protein and thereby causes the cancer. Okay, and ClinVar and NavMat positions also. So this is how the evidences are provided. Like I am saying, tier 1A and pathogenic, you can see the assessment as per the actionability. It gives you a criteria as well as evidences. So these are evidence backed up, means this will be either from database or publications which are well curated and quality checked. And same applies for pathogenicity also. And you can apply this one as things. And then from the actionability perspective, we have 3 sections. One is treatment. Okay, what are the drugs? Okay, so the first thing, whether that is sensitive or resistant or not established. What is the evidence guidelines, whether it is approved or it is at very clinical trial. So the evidence level as per the things. And also indication wise, you can look for specific cancer or you can change it to all cancer type also. So say for example, I'm looking for a NPM mutation. Okay, I know that it is there in AML, but I want to check what if is it present in any other cancer type. So then you can always check in all cancer types also. So that way you can check. And the clinical trials, you can filter down on the phase wise also, or by the location also you can filter it out. And also same way we provide the having this mutation, whether this is helpful for a patient with overall prognosis or response rate or overall survival. So all these informations are provided as backed by the evidences or the references in the prognostic outcomes. Okay, and as I mentioned to you, each of the references you can look at whether it is coming from database or is it from the curated content from us manually, or reference you added also in the lab. You can also add your own references which will also show the symbol. So that way, you know, end of the day where exactly the details are coming out. So when you have the variant list, you look at the variant informations, and then the last step, you are signing out the report. So you are looked at how many variants are there, and you are giving an overall comment of what are the guidelines says about it, what is the interactions, and then adding the markers, which has all this, like you can get a second expert interpretation from our colleagues from the expert interpretation team, as well as a report comment with disease summary, molecular function, and what does the CBio portal or cosmic database says about it in role of disease or incidence in such way. So like how much it is in AML, how much it is in particular population of patients. So all this information you get it through the CBio portal. Okay, so with all this, when you submit a final step, we generate QCA generates a report which can be customized with your lab logo as well as your the lab details, and we can customize on the what relevant information, patient ID, doctor's ID, what is the information you want, and with the signature we can generate this report, which will has editable comment page, what is the therapies which are approved, guidelines for and clinical trials which are there, and pathologist approval. So overall, this single page has instantly provided a relevant findings from your entire sample and detection status and what is the drugs which are approved in one single page. We can have multiple pages also which will have the details, but this page will tell you what is the overall status and how this is going to help with treatments and other findings. Okay, so this is how, and just to add, just to tell more, like IVDR approved, we got it, and like because it's only in Europe, a lot of people wanted to have this in Europe, and it's slowly developing. And also we are supporting one of the prestigious project like Danish Cancer Genome. So Danish Genome, they are doing whole genome sequencing for all the cancer patients for the last 2 years. So they're using QCI Interpret to support this initiative. Okay, and also we are supporting some pharma partners also with QCI, where they use our interpretation tool to select the right patient for their clinical trials or the drug designing. Okay, and then like when you say I can do it manually also, and there are a lot of tools available freely or paid ones also. How we different from this? So we have a publication published in Journal of Molecular Pathology. A lot of my colleagues are there who looked at hematological cancers and solid tumors, and they have went through a lot of variants and diversified classification. This 8 labs we selected who looked anonymously, and other labs like our colleagues, we checked through this is using supported tool like QCI, and there was a lot of difference. So after all the analysis, there were 149 variants, out of which 14 were discordant with the expert panel. Okay, after the software, but 41 variants reflected a disagreement between the human reviewers. There's 8 labs; they were never coming on the same page. So this is where, when I give a VCF file to you and all the 44 of you, you should not come as a different variant reporting. So this is where the tool like QCI, whenever you run it multiple people, the evidence backed up, it will show you the relevant information and makes more concordance amongst the clinicians or the researchers who are working on this. So this is where we can be more helpful for you, all the researchers or the clinicians, especially trying to help the patient. And then the most importantly also, all this tool, since it is a patient sensitive information also, this is HIPAA data security policies are there, HIPAA certificates and compliances, ISO certifications, all are in place, which make sure that the patient data is at utmost secure locations, used at without any harm. Okay, so this is where we can I would like to stop, and I'll be happy to take any questions.
Thanks a lot, Siva. Thanks a lot for your very very informative talk. Waiting for the question, I just want to asking about the software we can install locally, and we can run it locally, or we need to upload the data into cloud.
Yes.
To do the analysis.
Yeah, that's a very good question, actually. So there are 2 softwares which I presented. The CLC, let me share my screen again. Okay, can you see my screen?
Yes, I can see your screen now.
Yeah. So that's a very good question, actually. The CLC, the software which I mentioned, this slide. So the CLC is a local software. It runs locally, where you provide a fast queue. Either it runs on your local desktop or server or on the things. And the VCF which generates, you have to upload it to our server, which is present in Australia or in US. Okay, so that we have it as a lock and key model, where we lock it to your own system. Like when we give access to you to use this QCI, we make sure that only your information is visible to you. Okay, even I or any other colleagues from QIAGEN won't be able to see that. So in that way, we have the HIPAA and data security compliances afterward. So QCI, goes to the knowledge base which is sitting on the VCF file, goes to the knowledge base which is sitting on the server, brings the relevant information in the reportable format. That's how it is used.
Okay. So, and then my next question concerning about the configuration of my computer in local, in order to can run the CLC. How many CPU do I need? How many RAM memory do I need? And how many storage do I need?
So that's also a very good question on the point of how many samples you are doing. So per month, how many samples turnout you are doing? That's one thing. How big is your data? Are you running a small panels? how much GB, whether you are converting 100x, 200x into 2 GB, 3 GB data.
Okay.
So
Okay.
how much complexity is there? So those things decides on how much usage you need to do it. Okay. So those are the steps which tells you if any small panels with 16 GB RAM and 500 GB to 1 terabyte of hard disk, it runs very smoothly and it can run fast also. Okay. And our tools are ready to support whole genome and whole exome also, provided you run it on the capabilities. Also, you can upgrade it to, that is why like we go to server or in the cloud module to support this. Okay.
Okay. Okay. And then the VCF file, do we need to do the join call in order to have one VCF file for each run? For example, each run we have 48 sample per run. And then we need to do the join call to have the one VCF file containing 48 sample. Or we submit or we upload individual sample in the VCF file into the cloud.
Yes. So actually there are two ways also. So when you upload to QCI, you can submit as like all the different files. You can submit as single file also. If you can join it outside and do it as a single file uploading, or you bring individual VCF files for a single patient and you upload them as a same patient sample. So you can merge it within QCI itself. So that option is also available.
Okay. Okay. So you said that in the nearly the last slide, you said that you are waiting for the IVD for the software. So the IVD for the cancer diagnostic only, or for
yes,
every
cancer only.
Yes. For cancer only. Okay.
Yes. We already have we have IVDR certified in Europe. Okay. We as per the as I think Tanat was also mentioning, each country has different rules and regulations, and we have compliances of Europe. We have followed it, and we have got this QCI, especially for somatic, has got the IVR certifications in Europe.
So then the variant annotation for the somatic is follow the ACMG, or you follow what kind of guideline?
Yeah, so actually there are two things. For any somatic variant interpretation, there are two things. Okay. 1 second. Yeah, you can see, right? Actionability means what are the drugs approved, whether there is an approved drug available, whether there is a clinical trial. So that is called as actionability. That is provided for as per American Molecular Pathology or CAP or ASCO guidelines. Okay. The pathogenicity is standard as per the ACMG guidelines.
Okay.
So we get both of these for this variants.
Okay, perfect. Yeah, thank you. So and then we have the question from Dr. Thời. Please directly asking, turn on your microphone, please.
Yeah, thank you very much for your presentation. And I have a couple of questions about the criteria for variant report and some questions for actionability criteria. For variant report, I would like to know what is the threshold for the depth of sequencing, and also the thresholds for VAF. I saw that in your presentation, there are variants with only more than 100 reads. The depth is about 150x, and is it too low to report for somatic samples? This is my first question, and I will ask just the following questions after.
Yeah, sure. So thank you, Dr. for this question. But so technically, QCI is just an interpretation of the information. Okay, the one which decides the variant allele frequency (VAF) is through the secondary analysis tool, which is there like CLC Genomics Workbench. So we, based on the your wet lab, you can have 100x coverage, and what is the gene which is mutant, which is coverage. We can go up to 0.5x also, okay, 0.5% also, provided how much is your coverage. So if you have 500x coverage, we can go low as 0.5% also, okay. But if you have only 100x, so see, 1% means out of 100 cell, 1 cell has that cancer mutation. So do you think like, so our assay should be sensitive enough to detect that? So that's where we recommend you to run some of the positive control samples, okay, first for the assay validation, and have those mutations. Like there are some companies which provide you Horizon and other people, they provide you samples, cancer samples, which are known, having known mutation with particular allele frequency. So you run that and you standardize the wet lab, and you see whether it is same thing is detected at same allele frequency in the a secondary analysis like CLC Genomics Workbench. And also I will tell you, if we compare between Illumina and Ion Torrent and long reads like ONT, there will be a lot of difference.
Okay, so is there any also criteria for sample selection before testing? I mean that tumor percentage, for example. Sometimes
yes.
you know we have small samples with low tumor percentage, so it's
Yes,
quite
yes.
difficult.
Yes. Definitely, there are some criteria like minimum, so we say anything about 20% tumor content is always better to look for that, because it avoids the dominance of normal cells. And then also it is important to have the fresh, what kind of samples. If you are looking from FFPE tissue, again, that's another challenge we have. So these contents also helpful for that. So tumor content as well as the tumor DNA quality, these things are also very important parameters to look into.
Okay, so thank you. And my next question is that I saw in your presentation you use AMP and CAP and ASCO guideline for classification of actionability.
Yes.
But actually, you know, your program gained the IVDR approval from the EU. Why in this case you don't apply the ESMO guideline, which is more, you know, appropriate, which is more suitable for EU regulation in the classification of actionability? Because just continue, please.
Agreed. No, that is a very nice notice, nice point you have caught up. So actually, the classifications or the guidelines we follow from AMP, CAP, and ASCO. But the if I show you here in the next slide, where is it? So we have all the information from NCCN to ESMO, NCCN, and other data like ESMO, NCCN. You can see here, right? ESMO is there. Okay, NCCN, PDA, and we can filter by that also. So it's there. So that's already part of it. It's only the actionability criteria which are approved and followed worldwide is as per the guidelines what I told you, AMP, ASCO, and this one. Okay, and also currently we are in a transition phase. The whole world will go to VICC guideline system in coming next year. So we already providing customer VICC score and how it is helpful. So that will also be launched, I think, by this year end. All the people will move to VICC, and even QCI will adopt it. So that's how it will go. And just to answer your question in one line, yes, we have ESMO and everything. Only for the actionability perspective, the approved methodology is as per the AMP, ASCO, and CAP guidelines. So we have that standard guidelines used there.
Okay, thank you. So my last question is that for actionability criteria, classification, I saw that these are, for example, you know, like 1A, 1B, 2A. So I mean that this is just a general combined classification provided by QGenes, or you based on some specific classification. For example, for ESMO, there there is ECAT classification for actionability, and also for ASCO there's another classification. So just
Yeah.
back to a couple of slides, please.
Yeah. Actually, that's what we take all the things. It's not just one thing. Okay. So even if it ESMO says this one, it will be reported there. Okay, and we leave it to the end user to choose whether they want to go with ESMO or ASCO or anything else. Okay, so all the relevant informations will be there. If it is classified as 1A and ESMO has agreed and somebody else has disagreed, so that information will also be there. So if you click on this small triangle, right, what different people like ESMO, ASCO, everyone has told, that will be there as listed, what each person has told for this variant.
Okay, thank you very much.
Okay, I hope it answers your question. Thank you, Dr. Ryan.
Thank you, Siva. Thank you, Dr. Thịnh. So we still have so I let you take a break before several questions come to you, Siva. So
Yeah, sure.
The next question. So in Vietnam, they do some kind of sequencing, and then do they need to submit or upload the data, the raw data, to QIAGEN to have the final report?
No, that's what I was trying to tell you. You are
It's
not
not me.
uploading.
It's not me. It's from the other one.
Yeah. So that's what the first part is. You have the raw data that is in FASTQ format. So that is, you are not uploading it to any server or any cloud material. You are doing it locally on your laptop or in your own server. Okay. Then you are getting a VCF file. That is a file which has the variant from the mutation. Okay. But that doesn't have any patient information. But the thing is, the knowledge base means what is that clinical trials, ACMG, publications, everything is sitting in the server, which is there in the Australia or in US. So we have to protect that. So that is where we have a web-based tool, that is QCI. Okay, you use QCI to upload your VCF file and pull out all the relevant information from the knowledge base, which helps you to classify whether there is an approved drug, approved therapy, approved clinical trials, what is happening in there. So that information. So that step 2, this is where it will go to a approved server. But we also have, that's why I mentioned to you in the last slide, it is well HIPAA, ISO and certified, and a lot of people use it globally. And we make sure that data protection policy is strictly followed. So we both use up the sign of the agreement, where, even if you ask me, I won't be able to look into your data until and unless you provide access to me. Okay, so that's how the lock and key model works with you. So when, like in your institute itself, not everyone can visualize. Only the ones you have mapped to that tool can only visualize those data and information. And also, we don't use those data for anything else.
Yeah, thank you. So bạn
I hope this clarifies the answer. Yeah.
Yes, I will translate into Vietnamese for him. Yeah. Bạn Nguyễn Hoàng Huỳnh
Yeah,
Anh
please,
Phúc
please.
thì có nghĩa là data của bạn phải có một cái máy đó gì đó local, cái máy ở văn phòng của bạn đó, để bạn phải có cái cấu hình máy á, nó phải phù hợp với lại cái lượng data mà bạn giải á, để bạn có thể install cái software CLC đó, rồi bạn upload data của bạn lên vô trong cái CLC đó. Thì dĩ nhiên là cấu hình máy mình phải bự để mình đủ chạy, phải đủ lớn để đủ chạy cho cái data lớn của mình đó. Và dĩ nhiên là cài cái máy đó thì cài cái software CLC thì phải trả tiền đó. Rồi xong xuôi rồi bạn ra được cái file VCF file, xong rồi cái VCF file nó sẽ chứa những cái biến thể á, rồi bạn lại upload lên trên cloud. Nó lại có một cái software trên cloud đó, là như cái bạn Lưu Anh ở đây. Tôi xin lỗi là tại tôi không biết bạn Lưu Anh là ở đâu, bởi vì tôi hỏi cái bạn Siva cho nó chắc ha. Thì bạn Lưu Anh là viết lại chính xác rồi đó, cái mà tiến sĩ Siva nói, được không anh Phúc? Em nắm được không em? Rồi, so so the next question about the software, you said that you have the IVD in Europe. It's not yet, but it's coming soon. Yeah. And he asking that how about in Vietnam? We can using
Yeah.
it for the Ministry of Health to satisfy the regulation from the Vietnamese Ministry of Health or not? Yeah. So the question.
See, yeah, thank you. That's a great again. The looking at the perspective. So we had like each country wise, the rules and regulations are different. So that is where we are working on that. And also, see, there are every quarter we try to do some improvements. So we are working on that based on the number of samples. If more samples are there in each country, based on that we will take the priority. So we also have like in another few more months, we are trying to using AI convert into local languages also. Like Vietnamese, can you publish a report in Vietnamese? Something like that we are working out in backend. I won't tell that it will come this state, but we always working on new things. Yes, and we also want to have like a IVD for the entire thing, but only thing because it's limited, since we have different rules and different application forms itself to submit to each country, and their rules and regulations are different. It will take some time to prioritize and get that approval done. And also these are very they are not constant, they keep changing. So that is another thing which we need to make sure everything is in right place. Okay. And majorly because of few big projects, we were able to do it in Europe as a priority.
Okay. So the next question is concerning the update of your database. How often you update your database?
Yeah, that's a important and relevant information, because this is where you want a quality data, and as well as content which is trustable and which is up to date. Okay. So last week, if ClinVar has released something or clinical trials has done something, you get that updated everything. Okay. Even the publications also. Okay. If you yourself has published some data and it was released last week, then we make sure that we get the updated content within the 2 weeks of time. As well as every quarter, we do some feature changes as well as all the informations. But ACMG or ASCO or anything criteria has changed, we try to make sure as soon as possible, so that the latest information is brought to the end users.
Yeah. And then how to deal with the inconsistent, like for example, last year we annotate with the pathogenic, but this year we annotate with the lightly benign, and how come we can deal with the situation?
Yeah. So that's also you can re-index the sample. Okay. So your samples, you just by clicking on the last year sample, it will rerun the sample and it will tell you last year it was likely pathogenic, but due to this evidence, I make sure that what is that evidence is clearly highlighted in some color, so that it may upgrade your sample to likely pathogenic to pathogenic, or tier 2B to 1A. So that also we provide you the information on that. Okay.
Okay. So the last thing concerning about the in silicon prediction tool, like in the CLC software, they use the CARD, the CADD score.
Yeah.
However, in some variant, it's not a binary score, and there is software UDR, the prediction tool, such as the SIP or Polypen tool or Reveal.
Yeah. So we have 11 prediction in silico prediction tools, which predicts whether this having this we have Reveal, we have CAT score, we have Splice AI, and we have QCI interpretation also, and we have expert interpretation team. See, there is an oncologist, 200 of them sitting in US and Europe and till to Japan. They curate the content and they also look through the evidences specifically from oncology perspective and report it. So all this together helps us to predict this. We have, just to answer, 11 in silico prediction tools which detects whether this having this mutation leads to gain or loss of mutation, or leads to a deleterious protein or a gain of function. So this information is provided to you along with the expert interpretation also. So this is something which you won't get it anywhere else. Yeah.
So the tool is very divergent, and what is the response for each stone using? Where come from? you using the original recommendation, or you follow up the ACMG or link gene publication or whatever?
Yes. So we actually from the content wise, we look from the ACMG guidelines and the priority for pathogenicity, and the ACAP ASCO guidelines for actionability, as well as the clinical research data which is there. We provide you that information also, and they are categorized as a different standard or moderate or strong support. Okay, so based on that, the criteria will change and the priorities will also change. Okay.
Yeah, thanks. Dr. Phúc Đinh, are you happy? Are you satisfied with the answer?
Vâng, em hài lòng với câu hỏi ạ.
Ok, câu trả lời á, câu trả lời là em happy rồi đúng không em?
Dạ vâng ạ, em happy với trả lời tiến sĩ Silva.
Ok. Nếu mà cần em cứ hỏi thêm không sao đâu em. Đó, câu hỏi cuối rồi. Ok, so that's the last question. Thank God. Dr. Sita, like you have the oral exam.
No worries.
Thank
No,
God
this
for
is
your
because
time.
that's an important step. I will tell you, when we report the information, it should be benefit to the patient who is the suffering end, and we are all doing it from sample to clinicians to everybody is trying to help the patient. So it should go as a relevant information. I agree with all the questions what you have asked. Those are nice questions. Thank you.
Yes, because our student is more about clinical student, medical student. Yeah, so that is in that the question is very specifically. Yeah.
Sure.
So, chỉ muốn nhắn nhủ với bạn Nguyễn Đoàn Huỳnh Anh Phúc á, là cái IVD này là chỉ cho cái ung thư thôi. Cái germline thì không có, được không em? Đó, bởi vì là không phải cái IVD nào nó cũng là IVD cho cả cái software hết, mà nó chỉ có cái phần là cho cái phần report cho cái ung thư thôi, được không em? Đó, hồi nãy tôi có hỏi á, lúc đó tôi có hỏi là cái IVD này là cho cái gì đó, bởi vậy là mình cẩn thận cái đó, chứ không phải IVD là nó universal, được không em? Đó mà người ta cũng chưa có nữa, đó người ta cũng đang chờ. Đó. Rồi, thanks a lot for three audience, and once again all we together we would like to say thank you for your time as well as your very nice talk and information. Yeah. Không biết các bạn có hỏi thêm gì không? Chung hết từ đầu đến cuối á. Có hỏi gì không mọi người? Rồi, không hỏi gì hết thì mọi người thấy là mình học đó. Mọi người cũng ôn lại cái bài của mình ha. Đó, là mình học tách chiết rồi xong xuôi rồi tách chiết thì mọi người cũng biết cái mẫu, mỗi loại mẫu nó sẽ có khó khăn khác nhau mọi người. Đó. Ở đây mình làm là thực tế, đó mình muốn không phải dạy lý thuyết không. Đó là thực tế nó có như thế nào mọi người cũng thấy đó. Mình có tách chiết đủ loại mẫu hết trơn. Đó, mỗi cái nó có khó khăn riêng của nó. Đó, bởi vậy là mình giống như thông qua tại vì cái này nhà người ta sản xuất mọi người, đó là người ta hiểu rõ nhất chứ không có ai hiểu rõ bằng hết, tại vì người làm nghiên cứu thì chỉ tập trung vô nghiên cứu thôi, đúng không? Ở trong lâm sàng, trong thực tế mình đâu biết đâu. Đó, tôi không biết, tôi nói thiệt, đó là phải điều kiện như thế nào, đó có những cái là học hỏi rất là nhiều. Đó, thông qua mấy cái buổi nói chuyện này thì mình mới hiểu rõ được. Đó, tại vì nhà sản xuất mà, người ta sản xuất ra cái gì thị trường nó cần, chứ không phải tôi nghiên cứu trong phòng thí nghiệm không cái gì đó, và nó có những cái chuẩn tôi cũng không hiểu, không biết nó là gì, đúng không? Đó, ví dụ tôi làm nghiên cứu thì đâu có biết IVD nó là gì đâu, đúng không? Mọi người thấy là bữa nay người ta cũng trình bày ra cho mọi người thấy. Đó, rồi nếu mà có hỏi gì thì mọi người cũng thấy là trên chợ người ta cũng có đưa cái thông tin đó, cái địa chỉ để liên lạc rồi này, cái email đó để mọi người có thể hỏi thêm nếu mà ngại hỏi ở đây. Một lần nữa cảm ơn các bạn tham gia cái lớp này đó. Và mấy tuần sau nữa thì cũng sẽ có mấy cái sản phẩm khác nhau nữa mọi người. Đó, cái nào nó cũng sẽ có ưu điểm có khuyết điểm đó, thì tùy theo cái nhu cầu mình đang làm cái gì, cần cái gì thì mình sẽ tham gia để mình có kiến thức của mình để mình hiểu chung luôn mọi người. Đó, có cái nhìn tổng quát đó, Ví dụ như là tuần sau là sẽ có cái bên DKSH mà phân phối cái hệ thống PacBio là long read, đó là cái phần mà cuối cùng của cái bạn báo cáo thứ hai đó, cho thấy là PacBio ta cũng đang sử dụng để giải đó cho những cái sản phẩm dùng để chẩn đoán mấy bệnh di truyền, xét nghiệm gen đó. Rồi, người ta bên PacBio có long read và short read luôn nha mọi người, chứ không phải chỉ có long read không đâu. Cái short read của người ta là cho cái ung thư đó mọi người, vậy nó rất là chính xác, nó rất là tốt luôn. Tôi phân tích đó, mấy cái data thì PacBio data là định về, nhìn rất thích mọi người đó. Again, thanks a lot for your time. Cảm ơn QIAGEN nha, thanks a lot QIAGEN. Cảm ơn bạn Tùng rất là nhiều, đó đã hỗ trợ chúng tôi có một cái bài giảng rất là hay. Bây giờ cũng tối rồi, chúc mọi người có một tối ngon. Thanks a lot and good night for everyone. Good night Siva, Nathan and Chi. Good night.
Dạ, cảm ơn anh Lợi nhiều lắm ạ. Dạ.
Yeah. Rồi, chào mọi người ạ.
Dạ em chào thầy, em chào mọi người. Cảm ơn mọi người rất nhiều.